Thank you for the introduction. Good morning, everyone. This project is that I'm going to actually talk about is supported by AgriFutures Australia, and it was completed last year. So what I'm going to talk about is basically focusing on the gut integrity, which is one important aspect of the gut health in general. And in this project, we specifically focused on intestinal barrier function. And to give a little bit of a background, basically, when we have a situation that everything is normal in the intestine, a single layer of enterocytes basically covering the intestine, and these cells are basically packed together with a series of complex proteins called tight junctions. And these proteins, alongside with the enterocyte itself, they form a barrier that prevents the entry of harmful and unwanted materials like microorganisms and basically bacteria and toxins and other things. And at the same time, it allows a selective passage of nutrients and essential electrolytes and water and other things that needs to be basically entered the body. But there are various factors that actually impact the functionality of intestine. And specifically, if we look at things like infection or enteric disease and toxins and stress, and even nutritional deficiencies, it can actually impact and make the gut more vulnerable. And basically, we'll have a situation which we have a higher permeability and causes the intestinal inflammation. And from that, we would have a compromised health and performance. In this particular project, which was completed last year, we looked at amino acids as a nutritional factor, and stress stimulated by glucocorticoids as a model. But as a bit of a background again on the role of amino acids, why we chose that those particular nutrients basically is that they are known to be fundamentally important in various intestinal functions. And as you can see in this slide, for example, if we look at the intestinal inflammation, these amino acids can actually contribute or both contribute to the intestinal inflammation and also reduce, depending on the type of pathway that they would actually work on. So despite the abundant literature that we have on the role of amino acids in nutrition of the chickens, and a lot of basically research has been done in performance, microbiota, and all those factors, the specific roles of these amino acids in poultry for the intestinal barrier functions are pretty much untapped and basically understudied. So the hypothesis, basically the idea is that these amino acids promote protein synthesis, of course, in enterocytes, and they help to maintain the adequate expression of those critical proteins, tight junctions, and they also impact various cells like gaplet cells, Paneth cells, immune cells, and also microbiota. So this was the main reason that we looked at. Alongside the project, we wanted to see whether, actually as a main objective, we wanted to see whether the dietary protein in the concept of reduced protein diets and amino acids can impact intestinal integrity using a basically barrier dysfunction model. And specifically, we had interest to look at a few of those promising amino acids based on other literature that is available for other species mainly. And the overarching basically objective was to optimize the intestinal integrity and performance, of course, of those birds that had already basically been fed reduced protein diets. The very first one, the first experiment that we did, we specifically looked at the concentration of amino acid and protein. We published that work last year in Poultry Science, so it is available for anyone who is interested to read. But to give a bit of a detail of what that experiment was about, we created a fairly large, basically wide range of impact in terms of dietary formulation. We had a reduced protein diets in grow around 17%, compared to a standard protein sitting around 20.2% at the time. And we had also a higher concentration of amino acid in high protein diet, basically to give a wide range to see whether we see any impact associated with both concentration of amino acids and reducing the dietary protein, which industry is very interested on. So we used a leaky gut model because we needed to create a situation that the barrier function is actually compromised to see any effects of these amino acids. And we use a very basically widely used method of permeability, which is by FITCD, fluorescent isothiocyanate dextran. And then we took the blood and we measured the FITC in those birds. And this shows you that there was indeed an impact and difference between the high protein and the birds that were fed reduced protein diets. And in the birds that were fed reduced protein, we had an indication of higher basically concentration of FITC, which implies higher permeability. And there was independent and basically big impact, large impact caused by dexamethasone as a glucocorticoid with known impact on the gut functions. We looked at wide range of tight junctions and we did PCR assays and you know, they are available in that paper, but in general, basically, just in a nutshell, we showed some reduction of villus surface area in jejunum when in the birds that were fed reduced protein diets, and the expression of one of the key tight junctions, ZO2, was lower than the birds that were fed standard protein diets. And we also saw a profound impact of the model that we use on expression of various tight junctions. But what I want to specifically mention about this study is basically we found that in the birds that were fed reduced protein diet, a higher basically expression of sodium-dependent glucose transporter, which is one of the main glucose transporters in the animals, and we've hypothesized that this might be one of the main reasons, or at least one of the contributing factors to excessive fat accumulation in the birds that are fed with reduced protein diets. And that simply is because of the known association with the high concentration of starch as the protein is reduced, and excess availability of glucose. But in terms of the gut integrity, what it makes it interesting is that things like SGLT1, it actually can impact the paracellular permeability, as you can see in these slides. If it's overly basically upregulated, it can either by stress or certain nutrients, in this case, possibly high amount of starch available in those diets, it can actually impact the intestinal permeability. And what we think it might happen is to have a low-grade inflammation in intestine. So it is very interesting to investigate these links between nutrient absorption and intestinal integrity. Follow on from that, we had a study that we specifically looked at a few of individual amino acids that we thought might have an impact on the intestinal integrity of those birds. And again, we published that last year, and it is an open access paper. So we followed the same design as the previous experiment, but we had a fairly moderate reduction in protein of the diet, only to 19.2% in grower, and we looked at the grower diets when we wanted to see the basically permeability and other parameters. We added glutamine and glycine and arginine into the basically reduced protein diets. And we had 1% glutamine, 1% glycine, and 0.5% arginine, which brought the arginine to around 140% of Ross recommendation, which was fairly substantial increase. But we wanted to see whether we see an impact, particularly under stress-stimulated conditions. We saw an impact on basically FCR only based on the arginine, only in the diets that had increased arginine, but there was no impact associated with glutamine and glycine that we could observe. But it was interesting that for the body weight, the birds that were fed reduced protein diets had a higher body weight. And one observation and that I need to mention here is that in this experiment, we had meat and bone meal in our control diet, standard protein. So, and obviously when we reduce the protein, we all know that often the meat and bone meal needs to be reduced. So we think that there might be some impact associated with the amount of meat and bone meal that we had in the standard diet. Again, on the gut permeability side of things, we didn't see any significant, statistically significant impact, but there was certainly a tendency to improve the and actually reduce the gut leakage in the birds that had higher concentration of arginine, and this was associated with a higher expression of claudin 1 as one of the barrier forming tight junctions. That convinced us basically to do some more gene expression studies, to do more mechanistic studies to basically see whether there are potential for these amino acids to impact the intestinal inflammation itself. And we found that particularly for glycine, there seems to be a reduction. Actually, there was a significant reduction in one of the main pathways that is involved in the intestinal inflammation, which is by NF-kappa B. So, but we also found that we hypothesized that if we do these experiments again, and for further studies, we need to perhaps reduce the protein below the amount of what we basically investigated in this project to see a more explicit response. To give you some take-home messages, I believe that strengthening the intestinal barrier is an excellent opportunity to influence health and performance of broilers and in poultry in general, and that is through basically developing resilience to enteric disease. And also, we think that there is a link between the mechanism of nutrient absorption and intestinal permeability and integrity in general, which requires further research. And we actually established that the concentration of amino acids and the level of protein can impact intestinal barrier function. We've got a new project actually recently funded as a continuation of this project, in which we are looking at more amino acids and specific amino acids in broilers that are fed a lower amount of protein to follow up on some of these interesting results. And with that, I'll thank you again for your attention, and thanks AgriFutures for the opportunity to speak about this project. Thank you.